Hello, my name is Karolena Lein, and this is part one of the article presentation for charting cellular identity during human in vitro beta cell differentiation. So just to begin with, I'm going to go through the agenda. First, I'm going to review some background information that's going to make understanding the paper easier, as well as the questions to be answered throughout the paper, and a bit about the experiment done. Lastly, I will go through figures 1 through 3 and discuss the different conclusions made from them. So to begin with, a pancreatic islet is an area within the pancreas that has the endocrine cells, or the hormone-producing cells. These islets are also called islets of Langerhans, which can be seen below in that picture, with the beta and alpha cells being produced. So a pancreatic beta cell is what creates and stores the insulin within the body, and it will secrete this insulin in response to glucose in order to maintain the homeostasis in the body. So when an individual has type 1 diabetes, their immune system will destroy these beta cells. And one therapeutic solution is to convert these pluripotent stem cells into pancreatic beta cells for transplantation. A previous study, you can kind of see a picture of it below, uses stepwise differentiation and a combination of signaling cues to create beta cells in vivo. These cells were able to secrete insulin in response to glucose challenges, and they were able to restore metabolic homeostasis in a mouse model. But the paper that we're focusing on today doesn't focus on in vivo, but focuses on in vitro differentiation protocols for developing cell-based therapies for diabetes. But one issue with in vitro protocols is the heterogeneity of cells. So throughout differentiation, some of the cells will branch off and become untargeted cell types. Identifying these cells developed can really help to improve the efficiency of the overall process. So this can kind of be related to our first article presentation, the dynamic chromatin remodeling mediated by polycomb proteins orchestrates pancreatic differentiation of human embryonic stem cells, which also kind of briefly touches on cell heterogeneity, but it's in relation to polyhormonal cells. This is when a single cell has multiple cell markers. However, both of these papers focus on the identification of these cells that they're developed throughout differentiation in order to improve the overall process. So one is different cell types within the overall population, and then one is polyhormonal cells. So the questions that this paper is going to be asking and what they're going to be answering throughout these different experiments is first is using single cell RNA sequencing, are the states and identities of all cell types that are developed before and during in vitro beta cell production comparable to a human islet, as well as some previous protocols that this paper brings up. And are any of these cells polyhormonal? Next, do these cells maintain their identities and functionality over time, specifically the beta cell population and its glucose responsiveness? And lastly, can a cell surface marker be found that allows for isolation of the SC beta cells? These questions are going to be addressed throughout the article. So within the experiment, this paper utilizes high throughput single cell RNA sequencing to characterize the cell types. This is able to profile thousands of individual cells and has been previously used in characterizing cell types in adult and human embryonic pancreases, but no previous study has comprehensively determined the identities and states of all the cell types produced before and alongside in vitro beta cells. Another test done is a glucose-stimulated insulin secretion test, and this is used to test for that beta cell functionality over time. And also you're going to see these T-distributed stochastic neighbor embedding projections, which are used to sort of visualize the single cell RNA populations. There are also some other techniques that I will mention throughout this presentation and explain what they're doing and what they're going to be used for. The experimental protocols that the paper is testing are referred to as X1, X2, and X3, and these are derived and compared to the production protocols, which are V1, V4, and V8. This is what the paper refers to them as. There is a table in the very bottom that is able to go over more in details what those include. The control group for this experiment is native human islets. So this experiment will begin with sequencing 40,444 cells at stages 3 through 6 in the differentiation process, with two different protocols. The protocols are derived from the previous research protocols that I've mentioned, the V1, V4, and V8. So figure 1A shows the cell types and the percentages of each at the stage from the original reference. And figure 1B gives a diagram of the RNA sequencing protocol with the indrops. So in figure A, the cell populations are shown through different colors. And you can see that in sort of a pie graph of what percentage of the population they are. And they are identified through different markers. For instance, this light green marker is a PDX1 progenitor and has the PDX1 marker. And this purple marker is an SC beta cell and has the INS NKX1.1 and CHGA markers. As you can see in this diagram below, here are the major cell groups identified at each stage based on those markers. So in this figure in 1C, you can see the expression profile of genes and markers that are relevant to the cell types that were previously developed in cell differentiation. The genes that are more expressed have a darker orange color as opposed to the non-expressed genes with a darker blue color, which is seen on that slider sort of on the right with expression level and cluster. The larger the white circle is within these boxes show that the more percentage of the cell population is expressing this marker. So some genes of interest are the ones that we saw previously in figure 1 for cell identification. PDX1 is a protein coding gene that is necessary for pancreatic development and beta cell maturation, which is what we saw in that progenitor. NKX6.1 is a gene in the pancreas that is needed for beta cell development. CHGA is found within neurons and endocrine cells and is a precursor to the active peptides vasostatin, pancreastatin, and peristatin. And last, INS is necessary for insulin production and provides instructions, which is going to be found in the beta cells and alpha cells. And GCG is a protein coding gene that develops glucagon. So in this slide, you can see the projections from the T, S, and E for both X1 and X2 experimental protocols. The major cell types of interest are going to be the SC beta and alpha cells, as well as the SC East T cells. And then because non-endocrine cells became mostly heterogeneous in stage 5 and 6, that is considered one group. Outside of these larger populations, there are a couple smaller groups that popped up, such as the SST HHEX. And as you can see between both of these projections, the overall population ratios of the cell types vary between the different protocols. However, the gene expression that's seen on the left has a high correlation between both protocols. So the researchers can conclude from this that while the protocols change the population ratios, that the cell type identities will not alter. So this is a quick figure from the appendix, and it's just briefly overviewing the experiment done and comparing cells at stage 6 with embryonic stem cells and induced pluripotent stem cells. And this just shows that there's a high correlation between them. So it can be concluded that changing the stem cell lines does not have a drastic effect of the progression of in vitro beta cell differentiation. So when we move on to the second figure, this shows the experimental procedure to answer if the cells maintain their stability over time, specifically the beta cells. In figure 1A on the left, this just shows the procedure for monitoring cell functionality during stage 6 for the 5 weeks using RNA-seq, and also the GSIS test. Figure B shows the insulin response for high and low glucose that is challenged across 3 different in vitro differentiations. And figure C shows the stimulation indices that is in comparison to the human islet control group. So the cells showed increase in glucose responsiveness over the weeks. The stimulation indices in figure C showed that the islets are comparative to human controls, but that there's a larger amount of insulin secretion. So researchers can conclude that glucose responsiveness over time for these beta cells increase as these cells mature. So once the behavior of these cells' functionality is determined, researchers kind of go on to looking at the identity of the cells overall. So in figure D, you can see that the stem cells, alpha, beta, and EC cells are defined, as well as the non-endocrine cells that we previously have seen in figure 1. The vertical bar icon shows the relative population ratios of each of these cell types through the weeks, and you can see different populations increasing over time, or some populations disappearing, like the neuro 3G progenitors. Figure E, this shows gene expressions which are overlaid on that projection. And the cells that are INS positive, that you can see sort of in the top left, are not expressing GCG, which shows that the cells over time become locked into their cellular types and that they are not becoming monohormonal. So researchers can conclude that the cells are maintaining their identities and are not differentiating towards a progenitor or another cell fate at stage 6. So these figures overview Posueto time analysis of stem cell beta cells. So Posueto time is a measure of a cell's trajectory or the transcriptional change as it progresses through cell differentiation. This is a single cell analysis, and this is really useful because at each given point, some cells may be further along in the progression than others, and using this technique, it can be ordered as such. So figure H shows the identification of dynamic genes with immature and mature cells. So immature is going to be on the left side, and mature is going to be on the right side. And when you're further away from the center, that's the mature and immature genes that are being expressed. The fold change along the bottom is comparing the beginning of the end of the cell's trajectory along Posueto time. And as we move on to figure I, this shows cell expressions along Posueto time for selected genes of interest. So each of the dots represent a cell that is sorted. It can be seen that certain genes increase or decrease along Posueto time. But overall, the researchers conclude that the changes are very subtle throughout the stage 6. But we can see some things. As the beta cell moves along the trajectory, it shows markers of maturity with an increase in IAPP and HOPX. We also see that there is an increase in transcriptional regulation around the insulin gene INS because LDHA and IGF2 are decreasing along with Posueto time. So in figure 3, we're overviewing the SCEC cells, and they show low levels of insulin, TPH1 and NKX6-1 gene, but they lack the beta cell markers. Just a little side note, TPH1 is related to serotonin synthesis. So the discovery of these cells was not expected by the researchers. And in figure 3A, you can see the genes needed for serotonin synthesis, such as the TPH1, as well as other markers for enterochromaffin cells, are within the SCEC's expression profile in comparison to the SC beta cells, which have the INS gene. The expression profile in figure B also shows a higher expression of these related genes in SCEC in comparison to SC alpha and beta cell, as well as human pancreatic endocrine cells on the bottom and non-endocrine cells. So if we further explore these cells with immunofluorescence staining of the EC markers in figure C and D, we can see that these SCEC cells contain serotonin and co-expresses the markers TPH1, as mentioned before, LMX1A, and SLC18A1. So as such, the SCE cells are concluded to be enterochromaffin cells. Figure E is also immunofluorescence staining of graft tissue after 6 weeks post-transplantation, which shows that these SCEC cells survive after transplantation. So endochromaffin cells secrete serotonin to control gastrointestinal neurons, and this is in response to multiple types of stimuli, such as mechanical forces. And this really helps with mucus production and sensory transduction within the gut. So these EC cells are normally found within the gut, and they're not normally part of a pancreatic islet. However, some studies have found that EC cells are within very rare EC tumor cells that can be found within the pancreas that are serotonin-producing tumor cells. So that kind of gives a reason to why they may be showing up in the study. Thank you so much for listening to part 1 of this project, and my partner will be working on part 2.